Okay, everyone. Let's start. Today's topic is mainly about the human immune reconstituted mice in translational medicine, challenges and solutions. We will mainly talk about these problems we are facing in application of in using the human immune reconstituted mice, and how do we solve these problems. And I am Huiyi Wang, the principal investigator in R&D department. And I am in charge of the immuno-oncology mouse models development. Okay, so many of us will be interested in what is humanized mouse models. Actually, this is humanized mouse means broadly refer to mice expressing human genes like transgene to gain relevant insights in the in vivo context for the understanding of human-specific psychology and pathology. While many basic biology insights have been obtained from mouse studies like B6 and biopsy mice, there are still many limitations to these mouse models when investigating human biology. Because there is still existing the human mice differences. There are components of mouse biology systems that are inconsistent with the human cord patterns, particularly the immune system. So, humanized mouse models have begun to fill this gap. In this webinar, we talk about humanized mouse models, mainly about the human immune reconstituted mouse models, which have the functional human immune cells and the tumor cells. And the humanized mouse models have begun to fill this gap and have become important preclinical tools for biomedical research. The use of humanized mice may facilitate the application of treatments in the medical industry. So, and how do we construct the humanized mouse models? Actually, one of the method to construct the humanized mouse model is transplanting the human stem cells and into the highly immune deficient mouse models like NCG mice, and it can develop a human partially develop a human immune resistant system. So, and we can use these preclinical mouse models to translate to humans and imitate the human disease like cancer and autoimmune diseases. When we want to construct the human immune reconstituted mouse models, we always want to use highly immune deficient mouse models, because if the mouse have a normal immune system, it will engulf the HSC cells, human HSC cells. So we need to so Gempharmatech has developed NCG mice. One NCG mice is the best mouse models for constructing a human immune reconstituted mouse model, because the background and the gene and strategy. The only mice have a deficient macrophage and DC cells. Also, the PRKDC gene the lockout of PRKDC gene will lead to the absence of T cells and B cells. And the L2RG, this gene is a common gamma chain receptor for L2, L4, L7, L9, L15, L21. So when we delete it, the L2RG gene. It means we block this cytokines ligand pathway, and it will lead to the absence of NK cells. And NCG is developed by Gempharmatech, and NCG is exclusively distributed by Charles River Laboratories in North America and European markets. And we also developed many next generation NCGs based on Gempharmatech's NCG mouse models. This figure shows the differences between the normal mice, nude mice, no-skin mice, and NCG mice. Actually, the white-type mice, like the node mice and the white-type B6 mice, they have a normal immune system, normal functional T cell, B cell, NK cell, macrophage, and DC and dendritic cells. And compared to the white-type mice and the biopsy nude, which is deletion of FOXN1 gene, and it will lead to the deficient of T cells, and but has a relatively normal B cell, NK cell, macrophage, and dendritic cells. And the non-SCID mice, the SCID means the severe combined immune deficiency, and non-SCID mice is we deleted the PRKDC gene based on the NOD background. So this mice is deficient of is absent of T cell, B cell, but has a relatively but have a NK cell and a deficient macrophage and dendritic cells. But non-SCID is not an ideal model to engraft the human immune cells, because it still has functional NK cells, and the NK cell will cytolytic the human immune cells. So the reconstitution of human immune system in non-SCID is relatively hard. And then the development of NCG mice based on the non-SCID, we delete the L2RG gene on the non-SCID. So this mice is the most highly deficient immune deficient mouse models in the world. One of mouse models in the world. It has an absence of T cell, B cell, NK cell, and has deficient macrophage and the dendritics, and which means it's an ideal model to engraft the human immune cells. And we have developed 20 plus next generation NCG mouse models to improve the engraftment options. Actually, usually we have two methods to construct the immune reconstituted mouse models. One is the PBMC NCG mouse model. PBMC is specialized immune blood cells with round nuclei like monocytes, lymphocytes, and macrophages. And PBMC cells developed in the bone marrow, but actually it's mainly located in the body's peripheral blood. And the PBMC cells, the PBMCs are isolated from the peripheral blood. How do we think? This is the workflow to construct the Huiyi PBMC NCG mouse model, and firstly we will extract the PBMCs and from the healthy people, and then the this PBMC engrafted into the tumor-bearing NCG mice, and then this is the PBMC NCG-bearing tumor-bearing mouse models. Using these mouse models, we can do some immuno-oncology studies, and the features of PBMC NCG mouse model is that it has faster reconstitution. It needs 1 to 2 weeks to reconstitute our immune system, and reconstitute more than 90% T cells, and the lifespan is about 4 to 7 weeks. Because of that, in the peripheral, the T cells, the lymphocytes, especially the T cells in the PBMC are T effector and T memory cells, so it can easily attack our easily attack the NCG mice. So the window period is relatively shorter, and it will when the T effector and memory T cell attack the NCG mice, it will easily lead to the GVHD, and which is the graft versus hoster, the hoster, to construct the PBMC NCG mouse model, we usually will conduct a PBMC donor screen in NCG mice, and so it's very important to have a PBMC donor bank, and we will and each donor will suppose 200 to 300 mice. And as we can see, there is the PBMC NCG reconstitution levels, and most of the from 2 to 4 weeks of age after the PBMC engraftment, there is the leukocytes proliferate at a relatively high speed, and but most of the leukocytes are the lymphocytes, CD3 positive T cells. And we use the PBMC NCG mouse models to conduct some immuno-oncology studies. We inoculate MDA MB231 breast cancer cells into the PBMC NCG mouse models, or the acute colorectal cancer cells into the PBMC NCG mice, and both of the MDA MB231 and acute PD-L1 highly expression tumor cell lines. So we treated the Tecentriq, and we treated the Tecentriq to test if the Tecentriq could inhibit the tumor growth in these mouse models. And as we all know, the Tecentriq is targeting the PD-L1, and PD-L1, when the PD-L1 binding to the PD-1 on the T cells, it will inhibit the T cells. It will inhibit the cytotoxic ability of our T cells. So their Tecentriq will so as we can see, the treatment of Tecentriq will enhance the cytotoxic ability of our T cells. And we can see, and the Tecentriq could obviously inhibit the MDA, the breast cancer cell, and the colorectal cancer cell growth in both in the PBMC NCG mouse models. Which means the PBMC NCG mouse model is a good mouse model to reconstitute the human T cells, and these T cells are also functional and could inhibit the tumor cell growth. Also, the second choice to reconstitute the human immune system is the HSC NCG mouse models. This mouse model is very different from the PMC mouse model because the cell source of these hematopoietic stem cells and the hematopoietic stem cells is a multipotent primitive cells that can develop into all types of blood cells, and it's nearly most of the hematopoietic stem cells are located in the bone marrow, and also found also found in the peripheral blood and the HSC cells mainly used in transplantation therapy. The HSC-NCG, the features of HSC-NCG mouse models is that it has we need a long time to reconstitute the human immune systems, like around 8 to 10 weeks, and the level of CD45, the human CD45 positive cells reaches more than 25% of PB after 10 weeks post-engraftment. And also the main immune cells is T cells and B cells, but the B cell is not a mature B cells, which means they couldn't secrete antigen specific antibodies to in this immune system. And the longest lifespan is more than 89 weeks, and which means that the PBMC, the HSC-NCG mice has a very long window period to conduct the efficacy study or to do some research, and the workflow to construct the HSC-NCG mice is that we will engraft the CD34 positive HSC cells into the irradiated NCG mice, which means the irradiated of NCG will construct a leash for the human HSC cells development and differentiated into different types of immune cells, and then we will around 10 to 14 weeks, when the T cell developed, we will inoculate or engrafted the cancer cells like CDX or PDX into the HSC-NCG mice, and then the HSC-NCG tumor-bearing mouse models is constructed. As we can see, the body weight of and the survival curve is very, the body weight is stably goes, and the survival curve is very stable, which means the lifespan of HSC-NCG is very stable and relatively long. As we can see, around 10 weeks, about 10 weeks post the HSC engraftment, the T cells gradually developed, and then the B cell, the percentage of B cells is gradually declined, but as we can see, there is a relatively low percentage of human NK cells in these mouse models. So this HSC NCG mice, human HSC NCG mice is mainly applied for the test articles that target to the T cells. And this is the T cell engagers evaluation study. As we use a Blincyto, this drug, and Blincyto is a CD3 CD19 bispecific antibody, but didn't have a FC domain. As we can see, when we inoculated the rat lymphoma cancer cells into the Huiyi and this is group 2 is treated with tree-specific antibodies which target the CD3 and the CD20, and as we can see, both of the tree-specific antibody or Blincyto could inhibit the tumor growth in this mouse model, which means this is which means the NCG have functional T cells, and these T cells could binding by the Blincyto and this tree-specific test articles and to inhibit the rat tumor cell in growth. And as we talked before, the PBMC NCG have a relatively shorter relatively shorter window period, has limited the window period to conduct efficacy study, and the Huiyi NCG have a relatively low development of NK cells and myeloid cells. To solve this problem, we developed to support NK cell development. We have developed the NK-IL-15 mouse model, and to support the myeloid and Treg cell development, we have developed the NCGM mouse model. And so firstly, we will introduce the NCGM, and we insert as human SCF L3 GM-CSF into the NCG and construct the NCG mice. This is the MR expression level in the marrow, now and spring, and we can see that in the KI mice, in the NCG mice, there is a human SCF, GM-CSF, and L3 expression in the NCG mice, but not in NCG mice. And also there is we also comparing the we are comparing the immune reconstituted levels between NCGM and NCG mice. As we can see, when we collected the CD34 human hematopoietic stem cells into the NCG, and we can see there is comparing to the NCG mice, there is the NCGM, the body weight of NCGM is relatively stable, and the GVHD score is relatively high, but the survival rate is very stable. Very stable in this mouse model. And we comparing the reconstitution level and the different and the cell different about the cell development in this in NCGM, and we can see the total leukocytes, and the percentage of human CD35 positive leukocytes is obviously higher than the NCG mice, and also including the much higher CD3 positive T cells and the higher CD19 positive B cells, and CD44 CD6 NK cells and CD33 myeloid cells, but all of these immune cells is boosted in the NCGM mouse model. And at the end point, we collected the peripheral blood, spleen, and marrow, and we found the same results. And the human CD3 positive T cells and CD33 positive myeloid cells are boosted and much higher compared to the HSNCG mice. But there is a decline of CD19 positive B cells, but actually the decline of CD19 positive B cells is acceptable because in the human peripheral blood, the percentage of human CD19 positive B cells is around 14 to 16 percentage. And there is, as we can see, there is a much higher development of T-Rex compared to the NCG mice. This is the percentage, and this is the cell count. So if your test article is tagging the expression on the T-Rex, you also can try these mouse models. We have tested the myeloid cells development and monocytes, granulocytes, and neutrophils, and all of these myeloid cells highly development in the HSNCG mice compared to the NCG mice. Also, we conduct anti-tumor efficacy in a lung tumor A549 inoculate tumor-bearing mouse models, and we can see in this IO study, we can see the PD-1 have can slightly inhibit the A549 goes in the HSNCG mouse models, but the combination of PD-1 and agent X. This agent X is the tacking on agent X. It will the MOA of agent X is deliberation of the MDSC cells. And we can see the combination of PD-1 and agent X could obviously has a obviously higher inhibit the non-cancer growth in the HSNCG mouse model, which means this mouse model is a good mouse model to evaluate that the test articles that deliberate that will attacking the MDSC cells. And to 12, and as we talked before, that PBMC-NCG mice has a problem that will lead to the very relatively shorter window period in this mouse model, and to know the window period as long as possible, we have developed the NCG MHC-DQ mouse models. Then the NCG MHC-DQ mouse model, as we know, actually the GVHD, the occurrence of GVHD because of the class 1 MHC molecule and class 2 MHC molecule to present the antigen to CD8 and the CD4 T cells. When the CD4 T cell and the CD8 T cell recognize the antigen, and it will proliferate in the spring and at a high speed and attack our mice. And to avoid this problem, and we have developed the B2M tail mouse models. The B2M is a subunit of MHC1, and but also B2M is a subunit of FcRn. So when we deleted the B2M, it will distract the structure of MHC1 and the FcRn, and which will lead to the slow down the occurrence of GVHD, but also will lead to the IgG half-life, will significantly reduce the half-life of IgG drugs, and affecting the occurrence of PK/PD evaluation. So to solve this problem, we didn't knock out the B2M subunits, but we knock out the alpha subunits, which means the H2K1 and but the alpha subunit is the gene knocker, the gene that H2KD and H2DB that will we will lock out the H2KD and H2DB on the NOD mice, and also the MHC2 gene IE, these three genes on the NCG mice, and the H2 related gene were locked out, which means the MHC1 and MHC2 molecules will be destroyed, and the integrity of B2M was preserved, and also the GVHD reaction was slowed to a maximum extent. So the experimental window period was extended, and the proportion of immune cells will be normal, and occurrence of antibody molecular evaluation was guaranteed. And this is the detection of mouse MHC1 and MHC2 expression in NCG MHC-DQ mice. As we can see, compared to NCG, the MHC-DQ mouse don't express the MHC1 protein, MHC1 and MHC2 protein, and the TBNK monocytes, neutrophils, and dendritic cells in NCG MHC-DQ were almost identical to those of NCG mice. And the body weight survival rate and the GVHD score after PBMC engraftment in the NCG MHC-DQ mouse, as we can see, there is obviously body weight decline around week 2, at week 2 poster PBMC engraftments, but the body weight of NCG MHC-DQ is relatively stable, and also the survival rate and the GVHD score, and which is the NCG MHC-DQ showed reduced GVHD symptoms compared to the NCG, and with no severe loss of body weight, no survival rate, and nearly no GVHD symptoms. And both NCG and NCG MHC-DQ mice were engrafted with the same PBMC cells in both, and we can see the immune reconstitution can be performed well, and the reconstituted cells, and nearly all of this the reconstituted cells are human T cells. Also, we conducted a PK study, and comparing to the NCG and the IgG half-life in the NCG B2M mouse models is really is very short, and but as we can see, comparing to NCG mice, and the IgG half-life in NCG MHCDQ mouse are nearly the same. Also, we conducted the efficacy study. Also, we used Tecentriq, this test article, and the mice were inoculated with the MDMB231 breast cancer cell line at the age around 6 to 8 weeks. And as we can see, the Tecentriq, the treatment of Tecentriq can inhibit the breast tumor cells growth in both NCG, in PBMC NCG and PBMC NCG MHCDQ mouse models. But around the day 20, there is a decline of body weight in the PBMC NCG group, and there is the body weight of PBMC NCG MHCDQ group are very stable, which means this mouse model, the PBMC NCG MHCDQ mouse models could also is a good model, is ideal model to conduct the PD-L1 test article efficacy and have a very stable life status and body weight. Besides the immuno-oncology studies, there is many other applications of humanized mouse models, including autoimmune disease and infectious disease. And in the second part, we want to introduce the application of humanized mouse models in autoimmune disease. And as we and this and the humanized mouse models of in RA as an important advance in the generation of preclinical mouse models of RA would be the establishment of functional human immune system, or some of the components in mice, in which they mount autoimmune responses and the clinical features of RA. For this purpose, human hematopoietic stem and progenitor cells, peripheral blood mononuclear cells, which is PBMC, or tissues like the fetal liver and the fetal derived hematopoietic stem cells, and were engrafted into the immunodeficient mice, and which lack the these mouse models will lack the ability to reject the allografts, and so it will enable stable reconstitution with human cells. I like features which is the green colored, and the drawbacks red colors. Of the 5 main approaches to development of humanized mouse model of the eye, firstly, transplantation of eye tissue or in vitro injection of peripheral blood lymphocytes or monocytal cell lines, and secondary and co-implantation of normal human cartilage and eye patient derived fibroblasts. Third is the injection of SFMCs from eye patients or other stimulated in vitro or routes. And fourth is that the inoculation of eye patient-derived peripheral blood mononuclear cells. And fifth, and the induction eye-like disease by viral or bacteria triggers immune deficient mice that have been reconstituted with human hematopoietic progenitor cells. And And also, and Gempharmatech has tried this method to construct the eye disease in the humanized immune to the his mouse models, and we found that the this way can and the injected of bacteria or viral triggers could induce the eye symptoms in the mouse model. And also, the second most autoimmune disease is the IBD, is the a regulation of the this is the occurrence of IBD, regulation of the intestinal homeostasis in healthy and IBD inflamed gut. A healthy intestinal epithelial barrier in the presence of TGF-beta and I and L2 will promote the dendritic cells and macrophages and to stimulate the generation of inducible Tregs, inducible the Tregs. And TGF-beta and L2 are markers that contribute to the generation of inducible Tregs and establish and maintain the tolerogenic environment in a healthy gut. On the contrary, the IBD-induced inflammation induced the intestinal epithelial barriers and secrete the TGF-beta, L6 and L8, stimulating these dendritic cells and macrophages to produce the inflammatory T helper 17, like the L-cytokine IL into L17A, L22, L21. And creating an inflammation focus and disease intestine. And the workflow to development the His IBD mouse is that we will firstly to construct the His mouse models, which engrafted the CD34 positive hematopoietic stem cells into the irradiated highly immune deficient mouse models like NCG or NCG or NSG. And we will select this successfully reconstituted mouse model to induce the IBD mouse models. Actually, these mouse models, this paper published that they use the TNBS to induce the to contests the induced Huiyi NSG. And we found that DSS also could successfully induce the IBD autoimmune disease in the his mouse models. And this is the the induction of experimental contests by TNBS or DNBS, and to assess the pathology and the immunopathogenesis of contests. And we could also to do some efficacy study, especially this macromolecular drugs in these mouse models, like infliximab. And the third autoimmune disease model is the SLE. The other measure, and actually there are two ways to construct the SLE autoimmune mouse model. And firstly is that PBMC humanized the SLE mouse models. It can better simulate the clinical characteristics of patients of SLE, about their lifespan, and as we all know that the PBMC engrafted into the NCG or other immunodeficient mouse models will easily lead to the occurrence of GVHD, so the lifespan and survival rates are significantly lower than those of the spontaneous or induced laparosprone mouse models. So which will lead to a narrow period for observation and or treatment. And the other problem in this mouse model is that as we talked previously, this mouse model could reconstitute 19%, now above 19%, 19 percentage of these immune cells are T cells, but as we all know, the occurrence of our SLE is not induced by T cells, but also B cells. So if your test article is CD20 or CD19, like this target, so this mouse models is not a good mouse model. It's not a good choice for your it's not a good choice. And the second mouse model is the HSC Princeton SLE mouse model, but Gempharmatech has the third way to construct the HSC SLE mouse models. The other HSC Princeton and the HSC Princeton humanized SLE mouse model irradiates the mouse before this modeling. And this can provide more space for humanized construction through irradiation or pre-treatment with chemical reagents. A previous study compared the efficacy, the efficiency of transplantation with irradiation, and found that the human immune cells could survive better than pre-radiation to 2 to 3 grade to non-skin mice before injected of human HSC. Also, this modeling method will better reduce the clinical features of human SLE. As we know, the HSC reconstituted mouse models could reconstitute B cells and T cells and other immune cells. So these mouse models may be much better than this mouse model. And this is the next generation pipeline of NCGs, and we have NCGM L15 and NCG free, and NCGM L15 could boost the NK cells and myeloid and T cells to development, and NCG free could help the HSC cells to differentiate it into the DC cells, and also the NCGL2 TG mouse to have the Treg development, and to help the RBC development in the peripheral blood. We have developed the NCG TPO and EPO mouse model. And this is our next general NCG pipelines. And that's all. This is today's webinar. Okay. Do you have any question or any problem? If not, we are done for today's webinar. And have a nice day.